Very good evening, students. So yes, in our previous class we have started cell biology, of which we have begun with cell the unit of life. So far what did we complete from this chapter beta? We have looked up into all the segments and what is the maximum questionnaire that is put from all the segments of this chapter, where we have completed prokaryotic cell in detail, and then from eukaryotic cell we have just begun there, okay, and we have completely discussed about cell theory, discoveries, different shapes of the cells and different sizes of the cells. And today we are going to look into cell wall, cell membrane, endomembrane system, where under endomembrane system we are going to cover up all the organelles which are included under endomembrane system. What are they, ma? Endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles. There is 99% possibility where we can expect a question from these segments of this chapter this year in NEET 2026. After clearly analyzing past 5-10 years papers, I am saying you, we can expect a questionnaire from these segments of this chapter. So listen attentively, ma'am. So here you see, first is cell wall. So cell wall, we know cell wall is the limiting boundary of what? Plant cell. But here we have to recall two kingdom system of classification where we have learnt in the biological classification. Two kingdom system of classification is given by whom, beta? Two kingdom. Carolus Linnaeus. Yeah, no issues. Okay. So two kingdom system of classification given by Carolus Linnaeus. What did Carolus Linnaeus done? All the organisms, be it prokaryotic, eukaryotic, unicellular, multicellular, photosynthetic, non-photosynthetic, he has placed all those organisms into one kingdom, that is Plantae, and rest all the organisms which doesn't have any cell wall, he has put that into what? Animalia. Okay. As such, when all the organisms which have cell wall are grouped in Plantae, we got what? Prokaryotic organisms, unicellular eukaryotes, and multicellular eukaryotes like fungi also. Everything is placed under one kingdom only. As such, when we have to discuss about cell wall, usually the cell wall of Monera will have what composition, beta? Most of the Monera. We know bacteria are the sole members of the kingdom Monera. Most of the Monerans will have what? What cell wall? Yes, peptidoglycan cell wall. Peptidoglycan cell wall with NAG units. Those are N-acetyl glucosamine units. Fine. Second, if we have to see the fungal cell wall. Fungal cell wall is made up of chitin. Chitin is again a polymer of NAG units. And then third is what, Nana? Algal cell wall. Algal cell wall is composed of galactans, mannans, minerals like calcium carbonate. Okay. And then comes the plant cell wall. Plant cell wall is abundantly composed of cellulose. Along with cellulose, we also have hemicellulose, pectins and proteins. Okay. So these are the compositions where you have to know. Take down parallelly quickly. Monerans, most of them they are having what? Most of them are having peptidoglycan cell wall. Few like gram-negative bacteria, they will be composed highly with polysaccharides and unsaturated lipids. And then fungal cell wall is made up of chitin, N-acetyl glucosamine units. Algal cell wall is having what? Galactans, mannans, minerals like calcium carbonate. Plant cell wall is having what? Cellulose, hemicellulose and pectins. Clear, right? Now we will be looking into the layers of the plant cell wall. Fine. So first, during the time of cytokinesis, how cytokinesis happens inside a plant cell? From the center, at the center first cell plate is formed. Fine. This cell plates will turn to what? Middle lamella. Fine. This cell plate from the center, it moves to what? Periphery. From center to periphery. So this is what? Centripetal mode of cell wall formation. As such, the initial cell that is formed between two cells after karyokinesis is what? Cell plate. Cell plate is mature to what? Middle lamella. Middle lamella is composed of calcium pectate and magnesium pectate. Important composition, ma. You have to remember. Middle lamella is composed of calcium pectate and magnesium pectate. Middle lamella will stand as a common wall, common cell wall in between all the different plant cells. Okay, common cell wall. Middle lamella is the first formed layer composed of calcium pectate, magnesium pectate, and it is formed in between all the cell wall. It is the common layer between all the cell wall. Ma'am, repeat fungal cell. Fungal cell wall is composed of chitin beta by N-acetyl glucosamine units, NAG units. Okay? Yeah. Continue. So, middle lamella composed of calcium pectate and magnesium pectate. So, in fruits if you take this calcium pectate, magnesium pectate, they will get solubilized to pulp. Okay? In fruits they get solubilized to pulp. Fine? In ripened fruits. And this is the common layer between the plant cells, first formed layer. Next, inner to the middle lamella, if I have to draw two cells, assume these are the two plant cells. And middle lamella is the common wall layer. Okay. So, next to the middle lamella, what is formed? Next to the middle lamella, the wall layer that is formed is primary cell wall. Always do remember, the cell wall layers are formed from outside to inside. First outer layer is what? Middle lamella. Next to the middle lamella, we have what? Primary cell wall. Primary cell wall. Primary cell wall is thin, extensible, meristematic. Okay? Primary cell wall is thin, extensible, meristematic, and it has just cellulosic cell wall. Cellulosic cell wall. What does this primary cell wall do? Primary cell wall is just single layered. It is just one layered. Next, at the time of maturity, what happens is, inner to the primary cell wall, secondary wall layers are formed. Okay, inner to the primary cell wall, what are formed, ma? Secondary cell walls are formed. This secondary cell walls can be 2 to 3 layers. Secondary cell walls will be in close proximity with cell membrane. Okay, next is secondary cell wall. Secondary cell walls can have suberin depositions. Suberin depositions. Fine. They are very thick. They can be 2 to 3 layered. Fine. So, with the growth of this secondary cell wall, yes Abdul, any doubt? Yeah, with the growth of this secondary cell wall, what happens is, as primary cell wall is thin and extensible, it is fragile, it can break the primary cell wall as well. Okay, and primary cell wall is composed of parenchymatous cells. Parenchymatous cells. Clear, right, ma? Any doubt anywhere? Now, once again, quick repetition about cell walls. The very first layer that is formed during cytokinesis of a plant cell is what? Cell plate. Cell plate matures to what? Middle lamella. So, the division is from center to the periphery. As such, middle lamella is formed. Middle lamella is the first formed layer. It is made up of calcium pectate and magnesium pectate. It is the common wall layer between the cell walls. Okay. Next to the middle lamella, towards the inner side of the middle lamella, what is formed? Primary cell wall. Cells of primary cell wall are parenchymatous. They are thin, extensible, meristematic, and they are made up of cellulosic cell wall. Next, inner to the primary cell wall, we have 2 to 3 layers of secondary cell wall. This secondary cell wall will mostly have what? Suberin depositions. with the growth of the cell and secondary wall layers, the primary wall layer is breakable, it is fragile. Okay. Now you have to know from cytoplasm or from the cell membrane, if you see, from cell membrane to the periphery, from cell membrane to the periphery. What is the wall layer composition? Closer to the cell membrane, what we have, ma? Closer to the cell membrane, we have secondary cell wall. Closer to the cell membrane, secondary cell wall. Next to the secondary cell wall, we have primary cell wall. Next to that, we have middle lamella. Okay. Next to that, we have middle lamella. And from periphery to the center, this will be the order. Okay. This order you have to remember. And how the cytoplasm of two plant cells are connected, if that is the question. Cytoplasm of two plant cells, how they are connected? By plasmodesmatal connections. Cytoplasm of two plant cells are connected by what? Plasmodesmatal connections. Plasmodesmatal connections will have common layer and they are made up of what? Desmotubulin protein. Desmotubule. Okay. So, what are these? Plasmodesmatal connections. Plasmodesmatal connections. Made up of what tubules? Desmotubules. Desmotubules. Clear with the cell wall, ma? Any doubts anywhere? No, right? Yeah. Next, we have to learn about cell membrane. Okay. The study and discovery of cell membrane has more elucidated after the discovery of microscope in the year 1950s. In the year 1950s, after the discovery of electron microscope, the structure of cell membrane is more elucidated. As such, many models were proposed on cell membrane or plasma membranes, of which widely accepted model is what? Fluid mosaic model. Widely accepted model is fluid mosaic model. Fluid mosaic model is proposed by whom? Singer and Nicholson. Singer and Nicholson in the year 1972. Fine. Singer and Nicholson has discovered this in the year 1972 and that is fluid mosaic model. First we will learn on what cell did they discover the cell membrane. On the erythrocyte, that is RBC, right? On the erythrocyte, that is RBC, where they clearly mentioned the composition of protein and lipid, wherein protein composition is 52% and lipid composition is 40%. Okay, protein composition is 52% and lipid composition is 40% and rest 5 to 8% will be what? Carbohydrates. Carbohydrates will be 5 to 8%. Fine, clear till here? Now we will clearly look into the composition of cell membrane. Okay, composition of cell membrane. Take down parallelly with the composition of cell membrane. Cell membrane is composed of lipids, proteins and carbohydrates. First is what? Lipids, proteins and carbohydrates. So you see here quite. First, lipids, how they are arranged, na? Lipids have what? Hydrophilic head and hydrophobic tail. Fine. Hydrophilic head is facing towards the aqueous environment. Okay. As such, you will be having, right, hydrophilic head and hydrophobic tail. This is the lipid bilayer which is proposed by Singer and Nicholson in fluid mosaic model. This is how it will be, right, ma? So here this is what? Hydrophilic head. Hydrophilic polar head we will say. Polar head. This hydrophilic polar head is facing towards what side? Aqueous environment. Fine. And towards the cell membrane, towards the inner side, what do we have? Hydrophobic non-polar tail. So this hydrophobic non-polar tail is protected from the aqueous environment by hydrophilic polar head. Yes or no beta? Hydrophobic non-polar tail is protected from the aqueous environment by hydrophilic polar head. This is the bilipid layer. How these lipids are arranged in the plasma membrane? By quasi-fluid nature. They are just floating. Okay. So this is quasi-fluid nature of lipid. Remember the term carefully. Direct NCERT line. Okay. So this is what? Quasi-fluid nature of lipid. Next about your proteins. Proteins, we have 3 types of proteins. What are they? Peripheral proteins, integral proteins and transmembrane proteins. Okay? Peripheral proteins, integral proteins and transmembrane proteins. Peripheral proteins are present on the either ends of the lipid bilayer. They can be extracted easily with the help of detergents. Whereas integral proteins will transpan 2 sides of the lipid bilayer. Okay? And they are embedded inside the lipid bilayer membrane. Okay? So that is why they are called as integral proteins, and their extraction is difficult when compared to the peripheral proteins. Okay, and transmembrane proteins can transpan any of the sides of the lipid bilayer, or it can also transpan both the sides. Okay, in this case, can I tell all the integral proteins are transmembrane proteins, but not all the transmembrane proteins are integral proteins. Okay, important point beta, point to be noted. All integral proteins are transmembrane proteins, but not all transmembrane proteins are integral proteins. Clear, right? Now it's about carbohydrates. Carbohydrates mainly help in cell to cell interaction beta. Carbohydrates mainly help in what? Cell to cell interaction. Okay, and how carbohydrates are present in the lipid bilayer? Either in the form of glycoproteins or in the form of glycolipids. Fine. Now here there is a statement. Quasi-fluid nature of lipid, I told you, right, that lipids will float. Quasi-fluid nature of lipids will allow the lateral movement of proteins. Okay. Quasi-fluid nature on the lipid bilayer will allow the lateral movement of proteins. Important point to be noted. Quasi-fluid nature of the lipids will allow the lateral movement of proteins. Okay, take down. Fine, right? So this is the structural composition which is seen in the cell membrane under fluid mosaic model. Okay, lipids will have hydrophilic head, polar head, and hydrophobic non-polar tail. Hydrophobic non-polar tail is protected from the aqueous environment by hydrophilic head. And how carbohydrates are present? Sugars are present either as glycoproteins or as glycolipids, which are being produced from Golgi bodies. Okay, and then we have 3 types of proteins. What are they beta? Peripheral proteins, integral proteins, transmembrane proteins. Peripheral proteins are present on the either sides of the lipid bilayer. Integral proteins are embedded inside the lipid bilayer, or it will transpan 2 sides of the lipid bilayer. Transmembrane proteins are either transpanning 1 layer or 2 layers of the lipid bilayer. So as such we can say all integral proteins are transmembrane proteins. Clear, right? you have understood the composition and the structural description of the cell membrane. Fine, from fluid mosaic model. Next, you listen. I will send you this PPT, listen. What are the major functions of the plasma membrane? The major functions of the plasma membrane includes, first is cell growth. Cell growth is purely dependent on the plasma membrane, because as cell size increases, which is the first layer that gets extended? Plasma membrane. If it is a plant cell, we have to consider first it is plasma membrane, then secondary wall layers, then primary wall layer will be extended. So, plasma membrane is involved in cell growth, then formation of intercellular junctions. Intercellular junctions, I already told you, plasmodesmata will connect the cytoplasm of two plant cells. For that plasmodesmatal connections, pits are present in the cell wall, okay, pits, those small pits are present. From that pit fields only plasmodesmatal connections will happen, which are aided by what? Plasma membrane. Then secretions, what are the secretions which are produced by the cell? Are produced from what? Cell membrane. Endocytosis and cell division. Cell division, that is cytokinesis, cell movement or cell division, okay, that is keenly promoted by what? Plasma membrane. First it is plasma membrane only, no, either cell furrow formation or cell plate formation happens from where? Cell membrane. Okay. So, these are the major functions of the plasma membrane. After this, you have to know the transport that happens in the plasma membrane. There are three modes of transport, beta, take down parallelly, three modes of the transport. First is what? First is passive transport. Second will be active transport, and third is bulk transport. Bulk transport. So, you have to know the difference first. Listen, passive transport means what, ma? Movement of solvent, be it water, okay, movement of the solvent from high concentration to low concentration. When water is moving from high concentration to low concentration, is there any energy required? No. Okay. So, no energy will be required for such transport from high concentration to low concentration. As such, we have three modes of passive transport. One is called as simple diffusion. One is simple diffusion. Second will be facilitated diffusion. Facilitated diffusion, and third will be osmosis. These are the three modes of transport from high concentration to low concentration without the requirement of any energy. Then if you have to take active transport, active transport is movement of molecules from low concentration to high concentration. When movement of molecules is from low concentration to high concentration, definitely there is a requirement of energy. So, energy is consumed and the energy is cellular energy, that is ATP. Clear, right, ma? Next is bulk transport, that is bulk movement of molecules, where molecules will move in groups. Okay. As such, we have movement of liquid molecules, movement of solid particles, okay, into the cell and out of the cell. We have movement into the cell and out of the cell. Movement of molecules into the cell, okay, first, that can be endocytosis. Endocytosis is again of two types, beta. Endocytosis is again of two types. It can be pinocytosis and phagocytosis. Can be pinocytosis and phagocytosis. Fine. Out of the cell is called as what? Exocytosis, which is also called as autophagy. Autophagy. So, here if you see, one by one we will learn. First is what I told you, passive transport. Passive transport can be through simple diffusion, facilitated diffusion and osmosis. Simple diffusion means movement of particles will be diffusing, like how air gets diffused. Okay, so most of the gaseous molecules and few of the neutral solutes will enter the cell through what? Simple diffusion. What are the molecules that follow the simple diffusion, beta? Yeah, gaseous molecules and neutral solutes. Gaseous molecules and neutral solutes. Fine. And next is what? Facilitated diffusion. The movement of molecules will be facilitated either by carrier proteins or channel proteins. Here you can clearly see in the image, you can clearly see how the movement of molecules from high concentration gradient to low concentration gradient, either through carrier proteins or through channel proteins, okay, like glucose transporters, sodium potassium pumps, that comes under active transport, glucose transporters, and how water molecule moves, okay. So that is through what? Facilitated diffusion. Water molecules will move through protein channels called as aquaporins. So facilitated diffusion, best example we can take water molecules, and the protein channels involved are what? Aquaporins. Fine. Next is osmosis. You are learning osmosis definitions from childhood. What is that? Movement of molecule from high to low concentration through a semi-permeable membrane or a plasma membrane or a cell membrane is called as osmosis. So osmosis movement is widely known, right? See, right? Movement of molecules, any molecules it might be, from high concentration to low concentration through the semi-permeable membrane. Mostly ion channels, those are being pumped through what? Osmosis. Fine. So here what is simple diffusion? Examples, best examples include what? Gaseous molecules and neutral solutes which just diffuse through the membrane. Next, like water molecules, ions, protein molecules, they will be transported through what? Either channel proteins or carrier proteins, that is facilitated diffusion. Next, osmosis is via plasma membrane, okay, from high concentration to low concentration. So these three modes of transport are through what? Passive transport. Next is what? Active transport, that is from low concentration to high concentration through semi-permeable membrane where ATP energy is required. So here best example can be sodium potassium pump. Fine. Clear with active transport and passive transport, beta. Now we will look into the bulk transport. Bulk transport is where movement of bulk molecules or huge molecules into the cell. If those molecules are liquid droplets, high amount of liquid droplets entering into any cell, that is called as penocytosis. So we can also call penocytosis as cell drinking. Okay. Phagocytosis, where movement of food molecules, solid particles into the cell, bulk of the molecules, that is called as phagocytosis. Best example, this is also called as what, ma? Cell eating. Phagocytosis is also called as cell eating. Exocytosis, omitting the bulk of molecules out of the cell, is called as exocytosis, which is also known as autophagy. This is also said to be what, beta? Cell vomiting. Okay, this is also known as what? Cell vomiting. Clear, right? Took all the points. Transport across the plasma membrane is clear to you know. Any doubt anywhere? Yeah. Now we will move on to the endomembrane system. So look at it carefully. How you can describe endomembrane system as? All the single membrane bound organelles whose functions are coordinated are put under the endomembrane system. Endomembrane system you can see only in the eukaryotic cells, because we have already discussed in prokaryotic cells there are no membrane bound organelles. We can see membrane bound organelles only in the eukaryotic cell. So under endomembrane system what organelles are included, beta? Take down the definition parallelly. Single membrane bound organelles, single membrane bound organelles whose functions are coordinated. Whose functions are coordinated. Fine. As such, 4 organelles are included under endomembrane system. What are they beta? Endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles. Of course, we will discuss each and every organelle in detail, but peripherally I will tell you the gist of endomembrane system in the first framework itself. Endoplasmic reticulum are nothing but what? See, this is the nucleus. Can you see here, ma? Yeah. So, nucleus, the outer extension of the nucleus is what? Nuclear envelope. This nuclear envelope reticulum structures will be extended as such, forming what? Endoplasmic reticulum. Endoplasmic reticulum is nothing but the reticulum extensions, tubular extensions which convert into a network of reticulum of nuclear envelope. Okay, this tubular extensions of nuclear envelope which form that network of reticulum structures is endoplasmic reticulum. This endoplasmic reticulum can be of 2 types. What are they? Rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum will be synthesizing proteins, smooth endoplasmic lipids. These two are naive, which are just synthesized inactive particles, proteins and lipids. They are accepted by Golgi bodies. I will tell you which phase of Golgi is accepting and what is happening inside Golgi. Inside Golgi bodies, what happens? These molecules will get packed completely. Okay, and these packed molecules will be released as the vesicles which are rich in hydrolytic enzymes. Okay, and few particles, glycolipids, glycoproteins will be translocated to the intracellular targets and extracellular targets. And lysosomes which are rich in hydrolytic enzymes, this will start digesting the molecules. Inside each cells, biomicromolecules will be present, few dead cell organelles, dead cell constituents will be present. So, this lysosomes will be attaching to them, okay, and post digestion, as we have already learnt, 90% of plant cell volume is occupied by what cells beta? What cell organelle? Vacuole. So, those dead particles or the dead cellular constituents will be deposited inside the vacuole. This is the gist of endomembrane system. Fine. Let us first begin with endoplasmic reticulum. So, as we have discussed, what is endoplasmic reticulum? This network of structure, okay, tubular extensions forming out network of reticulum from the nuclear envelope. First, we will see how ribosomes are connected to this endoplasmic reticulum. Okay, we know ribosomes have 2 subunits. Okay, ribosomes will be having 2 subunits. Larger subunit is 60S, smaller subunit is 40S. Together they form, ribosomes, 2 subunits are connected by magnesium units. More the association of magnesium ions, more the connection will be there. As such, it is what type in eukaryotic cells? 80S type of ribosomes. This larger subunit of the ribosome is connected to the endoplasmic reticulum. This larger subunit is connected to the endoplasmic reticulum by what? Ribofourin. This is not visible. Ribofourin. This is a protein channel. Okay, by ribofourin, larger subunit of ribosome is connected to the endoplasmic reticulum. So, on those endoplasmic reticulum where ribosomes are present, we call it as what beta? Rough endoplasmic reticulum. Okay, and smooth endoplasmic reticulum are in close proximity to the nuclear envelope. They are also tubular extensions only, but then there is no ribosome over it. No ribosomes, those are smooth endoplasmic reticulum. So, the only function of rough endoplasmic reticulum is what, ma? Protein synthesis. Okay, protein synthesis, because they have ribosomes. Ribosomes are purely involved in protein synthesis. So, their only function is what? Protein synthesis. Whereas smooth endoplasmic reticulum, if you take, what are the functions of smooth endoplasmic reticulum? Lipid synthesis, and it will modulate or moderate the calcium ion concentration, which will regulate the muscle contraction and relaxation. Okay, calcium ion concentration which will regulate muscle contraction and relaxation. Yes, this will synthesize calcium ions, this will modulate calcium ion concentration, which will help in regulation of muscle contraction and relaxation. Okay, and then detoxification of drugs. Detoxification of drugs, and only in the animal cells, smooth endoplasmic reticulum is also involved in steroid synthesis. Yeah, steroid hormones or steroid synthesis only in the animal cell. RER in animals, in plants is only involved in protein synthesis, because ribosomes are purely for what purpose? Protein synthesis purpose. Done? Any doubts anywhere beta? No, right? And one more point to be noted, endoplasmic reticulum is also called as microsomes. Done, right? Next. And one more important structural description from your NCERT liner. This tubular extensions will divide the cytoplasm into luminal and extraluminal compartments. Okay, see, look at here beta. So these tubular extensions are there, right? These extensions, whichever inside space is there, this is called as luminal compartment, and peripheral cytoplasm, whichever is there outside this endoplasmic reticulum, that is said to be what? Extraluminal compartment. Okay, luminal and extraluminal compartment. One more differentiation to say in between eukaryotic cell and prokaryotic cell is, in eukaryotic cell all cell organelles are present, right beta? So they will be showing extensive compartmentalization of cytoplasm. Okay, extensive compartmentalization of cytoplasm. Fine, extensive compartmentalization of cytoplasm is observed, and this is about endoplasmic reticulum, smooth endoplasmic reticulum and rough endoplasmic reticulum. And then you have Golgi bodies. Now I told you that SER is releasing out the lipids, RER is releasing out the proteins. Which phase of Golgi is actually accepting them? Look at here, very good, cis phase. So these are the incoming transport vesicles. Small vesicle structures are released, right, from endoplasmic reticulum, right? These vesicle structures will be accepted by the cis phase. Cis phase is also called as convex phase, yeah, convex phase or forming phase. Fine. So you have described endoplasmic reticulum as what? Tubular extensions, network of reticulum structures. How can you describe Golgi bodies as? Golgi bodies can be described as disc shaped cisternae structures. Disc shaped structures like cisternae. They are called as Golgi bodies. So the vesicles are accepted by the cis phase, inside which packaging will be taking place. Formation of glycolipids, glycoproteins are formed. Like nothing but the sugar molecules will be added, like mannose, sucrose. They will be added to what? Lipids and proteins. As such, they will be packed. And they will be released through what phase beta? Trans phase. Trans phase is also called as mature phase. Yeah, mature phase or concave phase. Fine. Again, these packed molecules are released as vesicles. As they are being secreting from the Golgi, we can also call them as what? Secretory vesicles. Now we will see in what type of cells Golgi bodies are absent totally, in what type of cells Golgi bodies are extensively present. Okay, Golgi bodies are absent They are absent in cells like what? Sperm cells are antherozoids of bryophytes and pteridophytes. Take down parallelly. Antherozoids of bryophytes and pteridophytes. And they are absent in sieve tube cells. Yes, mature sieve tube cells, and they are absent in RBC cells as well. Mature RBC cells. Fine. In what type of cells you can see extensively present Golgi bodies? Golgi bodies will be abundant. Abundant in acrosome. First is acrosome of sperm cells. Root cap cells of plant. Root cap cells of plant. So, why are these extensively present in these cells? In the acrosome of sperm cell, in the acrosome of sperm cell, and in the root cap cells of the plant. Like, the major function of glycolipids is to synthesize glycolipids, glycoproteins, which will give that mucilaginous coating. Mucilage secretion will be maximum, which will aid in positive geotrophic movement of the root cap cells, and during fertilization it will aid in the proper movement of the sperm cells. Understood? This is the presence and absence of the Golgi bodies, and this is the major function of the Golgi bodies. You have to remember about the cis phase and trans phase. And here you have to take down two more points, beta. Just add on to that. Like, you can see unicisternal Golgi bodies, means only single cisternae will be present in fungal cells. Okay, in fungal cells. You can see what? Unicisternal Golgi bodies. Fine, single cisternae will be present. And in plant cells, there will not be extensive connection between the cisternae. Cisternae are unconnected. Okay, the disc shaped structures or cisternae are unconnected. This unconnected cisternae are called as what, beta? Dictyosomes. Important term, many a times asked in your exam. Dictyosomes. Fine. And next is animal cell. Animal cell, you can see extensive connection between the cisternae. Highly connected cisternae with cis and trans phases you can see in the animal cell. Fine. So, these are connected in the animal cell. Dictyosomes, unicisternal Golgi bodies and animal cells. Clear, right, ma? Next. So, everything is clear about Golgi bodies and endoplasmic reticulum, no? Now it is time for lysosomes. Very important. Lysosomes, we have many types. Those vesicles which are just released from the trans phase of the Golgi, these vesicles are called as primary lysosomes. Primary lysosomes. Primary lysosomes are rich in the hydrolytic enzymes. They are rich in what enzymes, ma? Hydrolytic enzymes. Fine. They are rich in hydrolytic enzymes. These are the primary lysosomes. Clear, right? Primary lysosomes rich in hydrolytic enzymes. These primary lysosomes, when they are adjoined with primary lysosomes, when they are adjoined with some digestive molecule. Primary lysosomes adjoined with some digestive molecule forms out to be what? Secondary lysosomes. Secondary lysosomes. Secondary lysosomes are also called as heterophagosomes. And this digestive particle is called as what, ma? Phagosome inside the cell. Okay, primary lysosome along with the digestive particle forms out to be what? Secondary lysosomes, which is also called as heterophagosomes. What are the hydrolytic enzymes that are present over here? Hydrolytic enzymes can be what, ma? Very good. And carbohydrases, and proteases and hydrolases and lipases, right? Yeah, these will help in the digestion of what? Biomacromolecules which are present inside the cell. Now here you see secondary lysosomes, heterophagosomes are clear, right beta? Now, when primary lysosomes, when they are connected along with some dead molecules or dead cell organelles, they form out to be tertiary lysosomes or autophagosomes, we will say it as. What, ma? Autophagosomes. Autophagosomes, which will help in what? Autolysis, which will conduct autolysis or autodigestion of the cell organelles, dead cell organelles. Okay, that is the reason these are called as what? Suicidal bags of the cell. Suicidal bags of the cell. Fine. And there is one more point to be discussed over here, that is secondary lysosomes will digest the particles, right? Digestive molecules will get digested and they will be releasing out small particles. Those small particles are called as what? Residual bodies. You are clear with all these terms, right? What are primary lysosomes? You can write later, I will send the PPT. Listen. What are primary lysosomes? Those vesicles which are rich in hydrolytic enzymes, those which are released just from the trans phase of the Golgi, from the concave phase of the Golgi. Those are called as primary lysosomes. When primary lysosomes are adjoined to a phagosome, what is a phagosome? Any digestive molecule. It can be any biomacromolecule. Okay, it is adjoined, so we are calling it as what? Secondary lysosome or heterophagosome. This secondary lysosome or heterophagosome, after the digestion, when small particles will be released, they are called as what? Residual bodies. Okay, and then you have what? Primary lysosome along with some dead molecule or dead cell organelle present inside the cell. After adjoining, it will be forming what? Autophagosomes. They will aid in what? Autolysis or autodigestion. Fine. That is the reason we will call these cells as suicidal bags of the cell, and after the digestion of the phagosome, small particles will be released. Those are called as residual bodies. 4 terms clear to you, right? Yeah. Now, these lysosomes after digestion, they will be releasing out some sap, they will be releasing out some excretory products, they will be landing or dumped into the vacuole. Okay, vacuole here acts as what? Dump yard of the cell in a plant cell. So here you see what happens inside a vacuole. We know in plants vacuole is said to be sap vacuole beta. Vacuole is said to be what? Sap vacuole. And we know it occupies how much percent of the cell? 90% of the cell. And the wall, the membrane which is present over the vacuole is called as what? Tonoplast. Tonoplast is the membrane. Now you listen. already there are many excretory products and toxins which are present inside the vacuole, yes or no? It is already rich with many solute particles. So, the concentration inside the vacuole is high beta. Concentration is high inside the vacuole. Whereas in the cytoplasm, comparatively the pH of the vacuole, here the pH will be high, means concentration is low, okay, low concentration, fine. Now, transport of particles, waste particles from low concentration to high concentration through a membrane is what transport? Active transport. Here pH will be low, here pH is high. pH is high and concentration is low. So, clear, right? So, movement of molecules from low concentration to high concentration through a membrane requires ATP energy. So, what type of transport is happening here? Active transport. So, through the active transport, all the sap and excretory products of the cell, high concentration, but after digestion in cytoplasm, whichever residual bodies are there, excretory products are there, those are being transported to the vacuoles. Understood? So, the from cell cytoplasm to vacuole, fine. Sap, excretory products of cell cytoplasm are being transported to vacuole. Clear, right? Any doubt anywhere? Fine, easy. So, as such we have many types of vacuoles. Those vacuoles which are helpful in buoyancy called as what? Gas vacuoles. Those vacuoles which are helpful in osmoregulation called as what? Contractile vacuoles. They are seen in unicellular organisms, be it in protozoans or monerans. What type of vacuoles we can see, beta? Gas vacuoles. For what activity? Buoyancy. And then we have contractile vacuoles for osmoregulation. Clear? Any doubt anywhere? No, right? Next. So, with this complete endomembrane system is done. Endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles. Clear with these four concepts. What are rest left from cell the unit of life is, we have yet to discuss with amembranous organelles or non-membrane bound organelles. Non-membrane bound organelles include what, ma? Of course, ribosomes, yeah, centrioles in the animal cell. Okay, centrioles in animal cell. And then we have to discuss about microbodies, which are single membrane bound organelles, tiny particles which are present inside the cell. And then we have to discuss about double membrane bound organelles, which include chloroplast or plastids and mitochondria, nucleus. In nucleus we have to look into the structure of chromosome, yeah, shapes of the chromosomes. Shapes of chromosomes. This complete concept we will deal in the next class. So, next class will be the last class for cell the unit of life, and then we will start cell cycle and cell division. Okay. So, that is all for today's session, beta. Thank you. Any doubt anywhere you can ask me, fine. And online students also, you can drop a message in the chat box and you can ask me, beta. Thank you.